Today we will be talking about the GATE XLK 2015 and the question number is 20, and we will try to solve this question which came from the topic of microbiology. So let us first read the question what it states. The question states that an E. coli strain is grown initially on glucose as the sole carbon source. Upon complete consumption of glucose following 12 hours of growth, lactose is added as the sole carbon source and the strain is further grown for 12 hours. Assuming that the E. coli strain has a functional wild-type lac operon, while which one of the following profiles is the most accurate representation of beta-galactosidase, beta-gal expression in arbitrary units? They have actually given four graphs, and this question, if you read carefully, it came from the topic of lac operon. Okay, so you see in the question it is written as functional wild type lac operon. For solving this question, first what you need to know is about the basics of lac operon. So now let us look into what is a lac operon. So lac operon. What does operon means? So this operon means there are certain clusters of genes which are grouped together under a single promoter. In these are actually found normally in the prokaryotic organisms like bacteria, and this whole gene will be transcribed into a single mRNA. That these group of genes which are clustered together in a group, they are under in a single promoter. They are under in a single promoter, and this whole gene will be transcribed into a single mRNA, from which the translation will occur and give rise to different proteins respective to the different genes. Okay, so this is what is an operon is all about. Why it is called as lac operon? Because we are talking about lactose operon. Means this operon will be getting activated only when this lactose is present and glucose is not present. So as we know that glucose is the main source of carbohydrate for our body as well as for the prokaryotic organisms like bacteria, because glucose is very easily can be break down into its metabolites which can be ultimately used in the glycolysis process. So that's why if glucose is present, then the life is very easy, that the glucose can be easily metabolized and it can be utilized in the glycolysis process. But the problem arises when the glucose is not present or glucose is present in minimal quantity, whereas the lactose is present or lactose is the main carbohydrate source for bacteria. So in that scenario, the bacteria wants to survive by utilizing that lactose and by breaking down it into the glucose. Somehow it wants to break down this lactose, because we know that lactose is a disaccharide which is formed by D-glucose and D-galactose. So somehow if the bacteria can metabolize this lactose into glucose, then it will serve its purpose. Mind it, this is only they will be doing when glucose is not present, but lactose is present as their sole carbohydrate source. Now what they will do? They will try to actually they have a operon where different enzymes which are required for this lactose metabolism is encoded, and that operon where these gene clusters are present is known as the lac operon. So if you see here in the left hand side, so this has different sites that you can see in this lac operon. So these genes which are colored in gray, these genes are actually responsible to form certain proteins that will actually help in the metabolism of lactose into glucose. So what are these genes? These genes are LAC-Z, LAC-Y and LAC-A. So this LAC-Y actually, not this LAC-A, this LAC-Y, this actually code for the permease enzyme. Okay, and this LAC-Z, this actually code for the beta galactosidase enzyme. Okay, again I am repeating that LAC-Z codes for, or if we express the LAC-Z mRNA, then it will be giving beta galactosidase, whereas LAC-Y will code for permease. So what is the function of beta galactosidase? This beta galactosidase is known to act on this lactose when present. If lactose is present, beta galactosidase, 50% of this lactose will be transglycosylated by this beta galactosidase, and this lactose will now be forming into its isoform, that is allolactose. Okay, this will be forming allolactose, and 50% of lactose will be hydrolyzed by beta galactosidase into galactose and glucose. Further, the allolactose which will be forming, the beta galactosidase can again act on this allolactose and convert it into galactose and glucose. So ultimately our aim, or the aim of the bacteria, is to produce this glucose. Okay, so that they can be utilizing this for glycolysis process. Then why they are forming allolactose? What is the role of allolactose here? So that we will be coming in the next slides. So this is all what the gene of LAC-Z is all about. But I also mentioned about LAC-Y, that is the permease. Now this lactose need to enter into the cell of E. coli. So that how it will enter. This enters by the help of this permease enzyme, which actually makes the lactose permeable through the cell membrane of the E. coli, so that it can enter into the E. coli. So that is the role of this permease enzyme. LAC-A codes for an enzyme known as trans-acetylase, but the role of the trans-acetylase is not fully understood. Okay, so this is all about the gene cluster that this LAC operon contains. Apart from this gene cluster, there are certain regulatory elements into the operon that you can see, and those are this operator, this promoter, and this cap site. So what are these? The promoter is the region where the RNA polymerase will bind and actually lead to the transcription of these genes that we are talking about, this LAC-Z, LAC-Y, and LAC-A. So in this promoter, the RNA polymerase will bind and actually carry out the transcription. What is the operator? The operator is known as a negative regulatory element. Why negative regulatory element? Because this operator site is partially overlapping with this promoter site. So if something which is called as the repressor, so what is repressor, LAC repressor, we will be talking about in details in the next slide. But this repressor, if binds to this operator, then the partially this promoter site is blocked, and the RNA polymerase now can no longer bind to this promoter, and the transcription of these genes are inhibited. So that's why this operator is known as a negative regulatory site. Whereas this cap binding site, this cap site that you see in the left hand part, this cap binding site is a positive regulatory site. Why it is a positive regulatory site? Because when this cap protein along with certain cofactor which will bind to this cap and make it activate to bind to this cap site, if it binds to the cap site, then it will actually help the RNA polymerase to bind more strongly to the promoter, so that more and more mRNA transcripts can be prepared. So it helps in enhanced transcription of these genes. That's why this cap site is known as the positive regulator or positive regulatory site. Now, how? So the question should come in your mind that how this operon is actually sensing the lactose and the glucose, amount of lactose or glucose, because we talked about that it will work only when lactose is present and glucose is not present. But we are telling this how this operon is actually sensing that whether lactose is present or glucose is present or absent. So let us see what is going on. So for that, two regulators will turn the operon on and off. So this, just like our switch which can turn on and off, so there are two regulators which will actually decide whether this lac operon will be in a on condition or in a off condition. So what are the two regulators? So one is the sensor for the lactose. Okay, so that is the lac repressor. So this lac repressor will sense the amount of lactose which is present in that E. coli. The lac repressor is this repressor that we were talking in the previous slide. So this lactose sensor, which is this lac repressor, this is actually, it will block the transcription to happen. But when it stops acting as a repressor only when this lactose is present. When this lactose is present, somehow this repressor loses its property as a repressor and this transcription will again goes on. But normally when this repressor is present and lactose is absent, then this lac repressor will bind to this operator region strongly and inhibit the transcription. So this is the lactose sensor that we have talked about. And what it is sensing? It is sensing basically allolactose. How? We will be talking in short time. But another sensor which is present is this glucose sensor. The glucose sensor is this catabolite activator protein which binds to this cap site. So this glucose sensor is this protein, catabolite activator protein, this protein. This green color protein is the glucose sensor. Okay, so this actually helps in activating the transcription of genes only when the glucose level is low. So mind it, if the glucose level is high, then this cap binding protein can no longer activate the transcription. So, and how it is sensing, what is the sensing molecules that it is actually sensing for knowing that what is the glucose concentration in the cell? It is sensing basically cyclic AMP. So this box that you are seeing here, so this is the cyclic AMP, CAMP. Okay, so this is the cyclic AMP. So this need to bind to the cap, then only it will get activated and will bind to this cap site and start the transcription process. So these are the two basic sensors upon which the lac operon operates. So the basic idea is, if glucose is present, then the bacteria need not to take care of anything, because the glucose is already present. It will metabolize that glucose and use it in glycolysis. But the problem arises when the lactose is present. When lactose is present, then only the lac operon that we are talking about comes into play. If glucose is present, this lac operon must be in a repressed state, must be in an inhibited state, where these enzymes will not require to take play any role, because glucose is already present. But if glucose is absent, then only we require these enzymes to act. For that we require this promoter. In this promoter, the RNA polymerase to bind and transcribe this gene, so that these genes can act on the lactose and metabolize into glucose. So this is the basic idea behind lac operon. Now we are looking deeper into what is lac repressor. So lac repressor is a protein which is encoded by a gene known as the LAC-I. So LAC-I is independently one gene which is found in its own promoter. It has its own promoters. This LAC-I gene is not present in this whole operon, but it is present somewhere nearby to this operon, and this gene is under its own promoter, this LAC-I's own promoter. Okay, and the interesting fact is this LAC-I is constitutively expressed or continually expressed, means this LAC-I gene will be always expressed continuously no matter what. So always in every situation you will have your lac repressor present in the E. coli cell. But whether it will go and bind to the operator and repress or activate the lac operon, that is under question. So the question now you understand. So lactose, if lactose is absent, then this repressor will be going and strongly binding to the operator, and as I have already mentioned, if this repressor binds to the operator, since the operator and the promoter some regions are overlapping, it will actually sterically hinder the RNA polymerase to bind to the promoter, and thereby actually if thereby actually not able to transcribe these genes which are important for lactose metabolism, because lactose is absent, we don't require these extra genes to for our means for the synthesis, it will be a cost of extra energy. But when lactose is present, what it will be doing is that this lactose is actually converted into an allolactose by transglycosylation, as we talked about. So there is this transglycosylation, 50% of this lactose is converted into allolactose. So this allolactose, why it is important? Because this allolactose can actually now allosterically bind to this repressor. You see, there is a allosteric site where this allolactose will actually bind. This will only happen when lactose is present and it is metabolized to allolactose, or this is transglycosylate to allolactose. Allolactose will bind to the repressor, and as a result, the shape of the repressor will change because of this allosteric binding. As a result of that, it cannot now no longer bind to, it can no longer bind to this operator region. Now this operator is free. Now the RNA polymerase can easily sit here and transcribe these genes. But if someone asks you whether this will actually transcribe these genes for a longer period of time, or this transcription is more means it's activated in a higher rate or not, that we will be coming in the next slide. Okay, so this is all we have talked about the lac repressor, which is the sensor of lactose. Now let us talk about the glucose sensor, or which is known as the catabolite activator protein. So this catabolite activator protein will bind to this positive regulatory site ahead of this promoter region that I was mentioning in one of my slides. Okay, it's in the front slide. So this is the promoter and this is the cap binding site where this cap will bind. When it will bind? It will bind only when there is low amount of glucose, because when there is low amount of glucose, then it need to metabolize the lactose so that more glucose can be present. But already if high glucose is present, it does not require these genes to get transcribed. So why it will go and bind there? So how it is inhibited when the high level of glucose is present? That should be your question. Then the answer is, when glucose is high, then there is an enzyme which is known as the adenylate cyclase. This adenylate cyclase convert the ATP, adenosine triphosphate, into cyclic AMP. Okay, but in presence of high glucose, it actually inhibit or inactivate this adenylate cyclase. So now this ATP can no longer be converted into cyclic AMP when high amount of glucose is present. So if cyclic AMP is absent, then it can no longer bind to this cap. Okay, so what is important is that for the cap to bind to this cap binding site, is this cyclic AMP should first bind to the cap, then only this cap will get activated and bind to this cap binding site. But here, if the cyclic AMP is not activated, then or it is not present, then it cannot activate cap, and cap cannot bind to the cap binding site. So as a result of that, in high glucose, the transcription there will be transcription because the lactose is present, but but the transcription will not be a higher rate of transcription. There will be a low transcription will be occurring if catabolite activated protein is not binding to this cap site. So clear? So now in summary, what we have talked about, that what are the four conditions that can be present. If glucose is present, lactose is absent. If glucose is present, then this cyclic AMP will not be formed, it will not be bind to the cap, and the lactose is absent, which means that this lac operon will not work, which will be the repressor will be binding to the operator region, cap cannot bind to the cap site. As a result of that, the transcription will be halted, there will be no transcription. Whereas when glucose is present, lactose is also present. Now when glucose is present, because of high level of glucose, cyclic AMP cannot be formed, or it cannot be converted from ATP to cyclic AMP. That's why the cap will not bind to the cap binding site. But now lactose is present. So if lactose is present, then it will convert into allolactose, and the allolactose will bind to the repressor. As a result of that, the transcription will happen. But the transcription will halt after a period of time, because low amount of transcription will be happening, because the cap is not binding to the cap site. Now if glucose is absent and lactose is also absent, then what will happen is that this glucose is absent, it will be forming the cyclic AMP, but lactose is also absent. So if lactose is absent, then repressor cannot be detached from the operator region. So as a result of that, there will be no transcription as well. But when glucose is absent and lactose is present, then there will be a higher level of transcription. So all this is actually summarized in this table. And now we can look into our question, and all the question which will come from this lac operon. Now I think you will be able to solve that. Now let us look again minutely into our question. An E. coli strain is grown initially on glucose as the sole carbon source. Upon complete consumption of glucose following 12 hours of growth, first, what they have treated is they have grown these E. coli cells on glucose. Okay. Then what they are asking, that upon complete consumption of glucose, the glucose they have completely consumed and no glucose is left over after 12 hours of growth. Okay, so the glucose they have given, first glucose has been consumed after 12 hours, and after 12 hours they have added lactose, and it is the sole carbon source. So means they are talking about a condition where glucose is absent but lactose is present. So they are talking about this condition, that glucose is absent but lactose is present. Okay, so what will happen? So the strain is further grown for 12 hours. Then it is given some time to grow. Assuming that E. coli strain has a functional wild type lac operon, so they have given that the lac operon is actually functional. Okay, which one of the following profile is most accurate for beta galactosidase? So they have given that this condition where glucose is absent and lactose is present. So what should be the scenario? The lac operon will work and it will transcribe this LAC-Z gene which codes for beta lactamase, beta galactosidase. Okay, so that is this beta gal. So they have asked that what will be the profile looking like. Okay, so the answer of this question is, because this use of glucose as a nutrient with high efficiency, the enzymes for metabolizing glucose being constitutive are always present in the cell. So that I have talked that why glucose is advantageous over lactose, because the enzymes of glucose is more easily constitutively present always in the cell, and glucose is much easier to metabolize than lactose. So if glucose is present, then there is no problem for the E. coli to grow. Thus when glucose is abundant, there is no advantage in making the enzymes for metabolizing lactose, just like what we have discussed. So consequently, the lac operon is repressed when the glucose is present in adequate quantities, an effect known as catabolite repression. So that happened when after these 12 hours, the first 12 hours, when glucose was already only the sole carbon source present. But after 12 hours, what they have said that when glucose supply falls after 12 hours, the repression is now lifted. As we have seen here, the repression is now lifted. Okay, so for the result of that, the beta galactosidase will now be able to express. So what will happen that initially up to 12 hours there was no beta galactosidase expression because glucose was present, no lactose was present. So the previously this condition was happening. So here what you see, the transcription is not there, so the lactate cannot be formed, beta galactosidase cannot be formed. So that's why the initially it will be 0. Okay, but after 12 hours, this when this glucose is not present but lactose is present, that is this condition, then your lactate will transcribe and the beta galactosidase will increase. So this will increase, but after a point of time it will again get saturated. Okay, so the answer of this question is this option 4. For your reference, you can actually look into these courses which is offered by NPTEL. The first course is of Functional Genomics by Professor S. Ganesh of IIT Kanpur, and the second course of Cell Culture Technologies by Dr. Moinak Das, IIT Kanpur. And this you can follow for looking into the details of microbiology, lacoperon, as well as the different microbial growth kinetics. Thank you.